Hello, my name is Marta Benito-Garzon and I work at Biogeco, a mixed unit between the University of Bordeaux and INRAE. This presentation will tackle the problem of integrating intra-specific variation into species distribution models. First of all, why it is important to consider intra-specific variation. From an ecological point of view, intra-specific variation is important because it influences ecosystem function. This is the main result of a meta-analysis comparing the ecological effects of intra-specific and interspecific effects. They evaluated ecological responses including changes in abundance and biomass, rates of ecological processes, and changes in community composition. The results show that intraspecific effects are often comparable to and sometimes stronger than species effects. From an evolutionary point of view, intraspecific variation is important because it allows organisms to persist in changing environments by adapting or acclimating to new conditions. Phenotypic plasticity occurs when phenotypic expression is flexible and can maintain fitness in either environment without, here in red and in pink and green, without changes in the genotype. Adaptive evolution involves changes in allele frequencies at loci that result in increased fitness in the new environment. Hence, the main sources of intraspecific variation can have different implications in population sensitivity to climate change and hence species distributions. Let's start with classical species distribution models that are based on the occurrence. So species distribution models are correlational models of low structural complexity that rely on the statistical relationship between species occurrence and local climate. Two of their main assumptions are that they consider that there are no differences among populations, neglecting the local adaptation and populations that populations may suffer in the past. As they are based on the correlation between the environment where a species lives and its occurrence, it makes a binary relationship with the climate, assuming that one species has the highest fitness in the places where it inhabits and zero fitness where it doesn't inhabit. So how to overcome these two assumptions? These are mainly done by what we call a group of models that we call DeltaTraitSDM, in which there is no genetic uniformity and there is fitness variation based on traits. And how this happens? By using phenotypic measurements from common gardens. So we can fit models with reaction norms of trait values related with fitness to predict trait spatial distributions. First of starting the talk on this DeltaTraitSDM, I would like to introduce you this scenario-based exercise in which there is a comparison between the species distribution model, which is classical, what we call no differentiation. Here, all populations are equally plastic, and there are no differences between populations. And when we predict what happens with different populations from cold in blue colors to warm populations in red colors, we see that there are no differences in the predictions. However, when we use hypothetical reaction norms, for instance, here there is local adaptation and all populations are equally plastic, we have already different predictions for all the populations. Same happens when other scenarios are proposed as high margin plasticity, high central plasticity, or high leading edge plasticity, showing hence that accounting for this intraspecific trait variation is very important and can change enormously the predictions of the species distribution models. So this is the outline of what I would like to discuss today. First, to introduce DeltaTraitSDM and particularly how they solve the problem of genetic and fitness uniformity across species ranges, and then talk about the trait sensitivity and the trait relationship with fitness that in the end are those that define the species ranges. And the third point is how this trait sensitivity changes across the tree life cycle to end in how to use this model for guiding assisting gene flow programs. So DeltaTraitSDM, which are the data that we use and how can we calibrate and validate these models? So they are based in common gardens networks planted all across the species ranges. And this is a very good example. This is probably the most extensive common garden that exists in Europe, the one for European beech. And we see the trials that have been planted in 2 different years, and provenance is covering all the distribution range of the species. In these trials, different measurements as survival, tree height, phenology, etc., can be used because they have been measured over years. So we have the common gardens and the different phenotypes, and we can compare also that we regress against the climate of the provenances, that is giving us an idea of the adaptation to climate in the past, and the climate of the planting sites where the trees have been living for years. So this is giving more an idea of the environment, so the plasticity. And then using this equation, so the phenotype, genotype, environment, and their interaction, we can have the trait values of fitness-related traits across the species ranges, or to delimit the species ranges. So let's see an example still with this beech. So we have the different reaction norms. These are not real reaction norms. And then we have how we can use these reaction norms for trait spatial prediction. Here is survival. This is a model based on survival where we have areas with low probability of survival, that is in orange here, and areas with high probability of survival here in green. And what we see, and this is what we see is that these areas are different from the species ranges, which are here in dash. So how this happens? So we could say that there is not a good validation model, but indeed what is happening here is that the survival is low. This is an effect of the fitness. And I will discuss this with a real comparison between species distribution models for the semi-species and DeltaTraitSDM for fagus, for beech. So we have on the top the habitat suitability calculated by a classical SDM, and on the left and on the right, the probability of survival calculated by DeltaTraitSDM, and in the bottom, predictions for 2070 and prediction on habitat suitability and the probability of survival. In blue, we have zones with high suitability or survival, depending on the model within the species ranges. In orange, high suitability or survival outside the species ranges, the current species ranges, and in green, low suitability or survival within species ranges. So the main message that we can obtain from here is that suitable area for predicted with the species distribution models is very good, is representative of what the species ranges is, which is normal because it's based on the occurrence and absence currently of the species. So of course it's very good. It's a bit circular. And then when we predict for the future, what we see is that there is an enormous decrease of this area. With DeltaTraitSDM, we have a less realistic suitability or probability of survival map. So the distribution is narrower than it is nowadays. When we predict, what we see is that it's keeping very similar. So let's go more in detail into DeltaTraitSDM. So we have similar predictions for the present and future scenarios, giving us a less alarming future for the species ranges. And then we have zones, the orange, with the species is not there now. But they are predicted as a very good place for survival. And this is the west part of the range where the species is considered to be native and is vigorously regenerating. So, of course, it's not included in the occurrence of the models, but there is there. So the models are predicting that survival should be good here. We have then another part of the range, which is the east part of the range, where survival is low in the prediction, The species is already showing high mortality in these areas. So this is one of the problems of species distribution models. If there is occurrence, the relationship with climate is one. And here we have a more nuanced, detailed information. So we have a fitness that is low, and then the prediction is that the species will have low survival. So it's more realistic, but we need to look at the trait we are analyzing. And here we come with the problem of the traits itself. So survival is very linked with fitness. This where we have seen is young tree survival, but we have other fitness-related traits that can be useful as well, which is vertical growth. That is usually the most, the variable that has been measured more broadly in common gardens. And we have radial growth. We have also phenology as bud bursts and leaf senescence. So if we look at these traits that are all of them related with fitness, we may have a surprise. The first one is that the climate variables that is more important for defining the sensibility to climate is different from all of the traits. So we will say that vertical growth, radial growth, and survival is more linked with transpiration and precipitation, whereas the phenology is more linked with temperature and latitude, which is photoperiod. So what happens when we do the predictions, the spatial predictions with these traits? What happens is that we have very different predictions. This is the young tree survival that we saw before. So we have in red areas very low survival, so the east and the southern part of the range. But if we go into the vertical growth and radial growth that are quite similar between them, we have that these areas are quite blue. That there is particularly the vertical growth, and the low growth is only happening in some parts of the mountains. And if we go into the leaf unfolding, so leaf flashing, what we have is that there is a very strong gradient from southeast to north, showing the differences in photoperiod. So how can we define the species ranges from this? We can learn which traits are more delimiting the ranges of the species in each part of the range. But if we combine them, for instance, here we have a model that combines leaf folding as a function of vegetation. Is vegetation as a function of leaf folding? So phenology is a covariable of the model. What we have is that those parts that in the growth model are predicted to be very good for the species, they are indeed decreasing the tree growth when we limit the growth by the phenology, by the phenological changes expected for the future. So this is giving us a more realistic picture, but also a more difficult one to interpret and very dependent on the traits that we used. And the picture is more complicated than that, because most of the trees that we have worked with, they are young trees or they are mature trees, but the sensitivity to temperature change over the tree life cycle. So we will need to consider different ages to be sure that what our models are correct. And this is an example on the left on how the thermal optimum changes across the juvenile stage of the trees. And this is simulation work. And with an empirical work on the right for three Mediterranean pinus, Nigra, Pinaster, and Pinea, in which there is have been calculated a phenotypic plasticity index based on tree height. And what we see is that for trees growing in common gardens at 4, 8, and 16 years, we have a decrease of this phenotypic plasticity with age. So there is something that changes over the tree life cycle. But the problem is that so far, we don't have many studies helping with that. So this is the number of species that have been studied in common gardens, in which we have at least 3 common gardens, at least 3 population tested. And here is the age of those trees measured. So we see that we start clearly when the trees are the best, when the trees are 1 year old, but then there is a lack of knowledge in the really first stages of trees. So we don't know how they can influence, for instance, regeneration or in the first years. So looking at this, we started a series of translocation experiments to calibrate DeltaTraitSDM at the very early life stages. And this is an example for Quercus suber, where we collected the seeds all around the distribution range of the species in Europe and North Africa, and we planted them under controlled temperature in climatic chambers, so at 3 different temperatures during several months. And we monitor germination and also measure what we call the post-germination traits to fit delta trait species distribution models. And this is the germination probability. Here I'm showing the differences between a prediction of 2080 and 2021. So red colors shows a decrease in germination probability, whereas blue colors show an increase in probability of germination. And what we see is that we can identify those areas. Here is the central Iberian Peninsula, where the species is predicted to have low germination. But carefully, because these differences are almost negligible, so in this case, the germination is not that affected by an increase in temperature. But that can change with other traits, and you will see later. I continue on germination here. These are a compilation that we did from many other studies doing the same translocation experiments from tropical and subtropical species, Mediterranean, temperate, and boreal. And we made the predictions as well. So we have, for temperate trees, we have 2 temperate trees here. This is Robinia pseudoacacia and Populus tremuloides. We see that an increase in temperatures, given that the humidity is maintained, will increase the probability of germination. In Mediterranean species, it's not that clear. We have 2 species, which is Pinus haldrichi and Ausculus hippocastanum, that are predicted to increase their germination. But as we saw before, Quercus suber is more constrained by germination. What is very important here is that tropical species, at least the 3 ones that we have, but we see here with the transfer distances that all of them are very similar. They are around the 0 transfer distance, or they are very well adapted to their climate. What we see is that they are predicted to decrease their germination with temperature, showing that they are very close to the thermal threshold that they can allow. So I don't continue with another traits, because you will have 2 other presentations here from Marion Carme and from Eduardo Vicente, showing our results for other traits and other species. So I will finish with the applications. How can we apply the results of these models for assisting gene flow programs? And this is an example in which the provenances have been clustered in seed provenance clusters regarding the conditions, the environmental conditions of the places. So we have here for an example for Picea abies and Fagus sylvatica. And we have the different clusters here in the present, calculated for the present, and then predicted for the future to optimize tree height. So we have that the clusters will change a bit in the current conditions already. So they are showing that the trees are already maladapted in some places. And if we go into a prediction for a climate change scenario for 2080, what we have is that the clusters need to change a lot if we want to optimize tree height. Particularly look at the Fagus sylvatica, to continue with the beech example that we started before. So we have that mostly one seed provenance cluster, which is the SPC1, is covering all the species ranges with some contribution with the SPC5 and SPC7, showing how we can plant the seeds to optimize tree height. To finish, I would like to set some conclusions on the delta trait, some positive conclusions, which is that they give a less alarming future for species ranges than SDM, and they provide biological information based on measured traits. as we show. So we know why there is a decrease in some part of the range and which are the traits that are more influencing this decrease. They differentiate the main processes behind the intraspecific variation, so we actually can know if one species is more plastic or is more adapted, and how can this change across the climate change. A positive part is they are correlational models which are easy to parameterize, and they can be calibrated for most commercial species for which common garden data exist for adult trees. On the contrary, the negative part is that the predictions are based on experimental conditions that may not be the ones encountered in the field. And we need several traits measured across different climates to really account for the fitness of one species and make the relationship with the species ranges. And to finish, a very straightforward use of these models is to guide assisting gene flow programs. For adult traits and adult trees, we have a lot of species that have been already calibrated. This is just an example, but there are more because there are several groups working on this DeltaTraitSDM, so just once an example. On the contrary, for germination traits and seedlings, there are only few. I show you here today some example for Quercus suber, and then you will have more examples from Abies avia and Quercus petrea, Quercus pubescens. This is the work that we are working on nowadays. Just to finish, I would like to thank, first of all, you for listening, and then all my colleagues and the funding agencies. So thank you very much.